Good morning, all. Today topic is plant tissue culture technique. So last class we completed about molecular biology. In molecular biology, about the genome structure, chromosome structure, gene expression, that means protein synthesis, transcription in eukaryote, translation and eukaryotes. So this is in about molecular biology, and apart from that continuation in molecular biology, the next topic gene regulation there and some of the restriction endonucleases enzymes. So they remains in molecular biology. Before that, another topic in this plant tissue culture and mm, molecular biology. In molecular biology, some of the portion almost to 70% portion completed. That's why I had to choose the plant tissue culture topic also. So we need to complete tissue culture, some of the portion also. That's why I have taken tissue culture. So let's see the tissue culture. What is tissue culture? What is tissue culture? The tissue culture in this process, in vitro culture of explant, culturing of explant. That means what happened in this tissue which is separated from the plant, mother plant, from the plant. We separated the tissue, not only tissue, and also we have to separate it from the tissue or cell or organs. So they're separated from the plant and taken into the test tube in vitro and in vitro cultured as a plant, cultured as a plant. So this process called as a tissue culture. Actually, how the plant grow? The plant to generally grow in outside the weather when the seed, when the seed germinate, the seed germinate, produce the plant, right? Then how the, sorry, right. Then how the seed formed by the fertilization, ovule, fertilized ovule developed as a seed. So the fertilization, that means not fertilization, fertilization embryo form. Embryo is a part of the seed. This embryo formed by the zygote, which is diploid. This diploid zygote by fusion of male and female gamete, both are fused as a result formation of zygote. Now the male gamete and female gamete, both male gametes are these male gametes develop from the pollen grain or microspores. Female gamete, female gamete in megasporangium or ovule. So the pollen grain part of the stamen, androecium, gynoecium. So these both are part of the flower. So these both are part of the flower. So the flower is the part of the plant. Flower is part of the plant. So we get the seed, we get the, to get the seed, to get the one seed, we have to wait up to germinating as a plant. It takes several years. And we will have to wait up to production of flowers. It also taken several years. and with mature take some time. After mature, after fertilization, zygote, embryo. So this is the time taking period. So but we get the plant in short period, and also within the short period we get number of plants. So due to this, we should adopt tissue culture technique. In tissue culture technique, we created all optimum temperatures, all natural weather in vitro. So what are the requirement for plant growth in general? In general, the plant growth requirement, they are the proper soil and moisture, minerals, they are all required. So we will have to provide, they are all in vitro process. So anyway, in vitro, after we created all these facilities, now this explant, actually what we separate from the plant to grow as a plant, that is called as explant. It might be cell, or it might be tissue or organ. So these are in vitro, in laboratory process, can develop into new individual plants. New individual plants. So this is the word introduction about the plant tissue culture, tissue culture definition, and some of the introduction. And the plant tissue culture, the first discovered by Haberland. This is discovered by Haberland scientist, discovered by Haberland scientist. So in plant tissue culture has a great significance in plant biotechnology, especially the crop improvement program. The term tissue culture may be defined as the process of in vitro culture. Just now I have explained the in vitro culture of explant. What is explant here? The explant any part of the plant in nutrient medium under aseptic condition. We have to create a aseptic condition also. However, in general, the tissue culture include the term the tissue culture as well as the culture, organ culture, and suspension culture also. So this is the similar to the any type of culture technique. So the plant tissue culture is the fundamental to most aseptic of biotechnology of plants. It is evident now that plant biotechnology is one of the most beneficial of all the sciences. The product of plant biotechnology are being transferred rapidly from laboratory to the fieldwork. Let's see how it is generated and how to transfer to the field. I will explain later. In this culture, the contributes immensely for understanding the patterns and respond. Some of the factors also involved in this. Let's see the what are the requirement for the plant tissue culture. Already told you for the plant tissue culture, we need aseptic condition. We created. I will explain how to create aseptic condition. We need proper explant, proper laboratory, and test tube, so on. Right. So the basic requirement of technique of plant tissue culture, laboratory organization, culture media, and aseptic condition. First, what are the laboratory? Let's take change. The main requirement, culture media, aseptic condition, laboratory organization. First, let's discuss laboratory organization. So how we organize laboratory, this is first taken. First, after laboratory, first we need to uh, we need to maintain proper laboratory. So, right. See, right. First, let's see laboratory organization. So, in a standard tissue culture lab, there must be a few basic facilities such as media, room for preparation. We need new media room, media room for preparation, media room for preparation and sterilization and storage culture media. First of all, we have a one specialized room. This specialized room for media preparation. While preparation, there is a get some microbes. That's why we need to sterilize them, sterilization, sterilization process. And after that, after sterilization, we storage the tissue culture, all tissue culture plants. So that is the first step. That is the first step. And second one, facilitate for washing of labwares and explant, so on. The washing labwares, that mean maybe it is test tube or the laminar airflow pot, indoor pot. There we need to clean. And we have enough space for storage of labwares. The third, we have the enough space for storage with labwares. And the fourth one, the culture rooms are incubators where the conditions of temperature, humidity, and light can be maintained. They should be all properly worked. Observe data and collection of area. These are the most important for laboratory organization. So, in laboratory organization, in laboratory organization, first we maintain the room, the proper clear room for media preparation, sterilization, after sterilization, storage. That's the first one. Now facilitate for washing of lab wares, explant, the so on. And also the lab have the enough space for culturing plant. And these culture rooms are incubators where conditions of temperature, humidity, and light, etc. This can be maintained. So these are the take into consideration to maintain the proper lab. So that is the first one. Now the already we know the culture media, culture media, culture media. Now how to prepare the culture media? How to prepare? Let's see how to prepare the culture media. Right. So the formation the media on which explant is cultured called culture media. It is composed of various nutrients required for proper culturing. Culture media that mean it is act like a soil in general. In general, what are the general requirement plant to grow? The plant to grow requirement to soil inside the soil nutrients. So they are all we provide. They are all in provide in form of culture media. So in this culture media, we composed of various nutrient required for proper culturing. Different type of plants and organs need different composition of culture media. That is based on explant. Number of media, number of media have been advised for specific tissue culture exam. Some important, the most important one, the most of the utilized for MS media. MS Murshige Krug, MS media, Murshige Krug media, or LS medium, or B5 medium. LS Lismargard Krug medium, B5 Gamborg medium, White's medium. But mostly we utilize MS Murshige and Krug medium. And in this media, in MS media, already told you, we added some of the nutrients. The nutrients may be organic, inorganic. Let's see in this from the organic side, organic nutrients, organic, organic nutrients, vitamins like B1, vitamins like B1 vitamin, B1, another name thiamine or peridoxin, peridoxin, neotic acid B3, neotic acid, and antibiotic like streptomycin, kanamycin also, streptomycin and kanamycin. These are the antibiotics also utilized for this organic supplement. And these are the organic. And if you take the inorganic, uh, inorganic or inorganic, the inorganic micronutrients, iron, iron, manganese, zinc, molybdenum, zinc, molybdenum, copper, boron, boron. They are the micro. Some of the micronutrients also required. The micronutrients are nitrogen, nitrogen, sulfur, phosphorus, potassium, calcium, magnesium. These are all, sorry, these are all macro, and these are micronutrients. Actually, they are required for the plant to grow. In general, how the plant get these nutrients, micronutrients or macronutrients in general, how they get? In general, they can get from the soil minerals in the form of minerals they can get. But in aseptic condition, we will have to provide all these micro and macronutrients as well as the organic vitamins, cytokines, sorry, vitamins and antibiotics, streptomycin and kanamycin. And apart from that, another important amino acids like arginine, asparagine also we need to add. Apart from that, here in organic amino acids, we add some of the amino acids also add, and also and inorganic are completed, organic completed, now carbon and energy source. So for plant growth, for what are the required? First of all, you need to know what are the general requirement for plant growth. General requirement plant growth, we should for development, they can prepare their own food material, right? How they can prepare own food material? Water, carbon dioxide, chlorophyll pigment, sunlight, prepare food material. Based on this food material, the energy obtained, ATP energy obtained. This ATP energy utilized for plant to grow, utilized for plant to grow. This is one of the requirement. And generally, and another side from the plant to grow, soil, already you know the water required. Here the water requirement for the plant growth. So inside the soil, the water, some of the minerals, dissolved in water. So with help of this water minerals also absorbed by plant. So this mineral may be micro or macro. So they are already be artificially provided in vitro process. So this one completed. And apart from that, some of the important vitamins also, amino acids required, they also artificially be provided to that. And so how we provide this sugar? Can in vitro process can prepare their own food material? Excellent can prepare their own food material. Excellent cannot prepare their own food material because already it is detached from the plant. There is no relation to the plant, not only plant, there is no relation to sunlight also. It is in laboratory. That's why we need to provide this carbon and energy sources also. And for tissue culture, for this carbon and energy source, here the most preferred carbon source is sucrose. And apart from that, this sucrose also we add to the media. Sucrose, sucrose, and other include lactose and maltose, galactose, raffinose, cellulose. They are all. And okay, they are the primarily involved. So okay, the water required, carbon dioxide required, and actually the in vitro process not utilize carbon dioxide, not utilize water. So water with minerals we added carbon dioxide with actually this carbon dioxide. No carbon dioxide, no chlorophyll cannot prepare food material. That's why already the prepared sucrase we added to the media to so that to get ATP energy, right? So, and for growing, the another importance are the growth hormones also. Another important growth hormones. We should provide growth hormones too. What are the growth hormones here? Auxins, auxins, cytokinin, auxin, cytokinin, gibberellin, abscisic acid, gibberellin, abscisic acid used occasionally. This is what regular process. They occasionally used this one. So regularly in normal condition used by auxin, cytokinin, and gibberellin. These are the growth promoters. These three growth promoters. Right. So this is the aseptic condition, sorry, culture media preparation. So first of all, actually, what we completed first, lab should be neat. First of all, we have the space, enough space lab, the proper room for media, for sterilization, for storage, all this. And anyway, after selected the proper media, we need to prepare the culture media. So for this culture media, first of all, actually, we add agar gel. After add agar gel, we add these type of inorganic nutrients like ferrous, manganese, zinc, molybdenum, like that, and organic vitamins are vitamins and amino acids. And apart from the growth hormones also, we need to add this agar culture media. So anyway, in a culture media prepared. And another important one in culture media, sucrose. We have to add the sucrose also. Sucrose. We need to add. We need to add sucrose also because the sucrose is the energy source which produce the ATP energy. That's why these are all requirements. So this is the culture media preparation. After culture media preparation, we need to provide aseptic condition. Because sorry, we need to provide we need to provide aseptic condition. Why? Because here, why we need to provide aseptic condition? Because uh, actually in laboratory there is a chance to attack by microbes. Or while the preparing media also mainly based on sucrose food materials like sucrose and nutrients, micronutrients and macronutrients and vitamins, amino acid they are. So due to this, mainly they can act as a substrate to microbes. That's why while we preparing the culture media, there is immense chance to attack microbes. That's why we need to sterilize. We need to sterilize. We need to create aseptic condition to the in vitro culture media. So how to create a aseptic condition? To create aseptic condition, uh, first of all, the maintenance of aseptic condition is the most critical and difficult aspect in vitro culturing experiment. Because why? Because you have taken the proper enough ventilated the spacious laboratory to grow culture. Already you have selected, you have did that, and in second step, already you have prepared well culture media with micro and macro nutrients, sucrose, agar media. But if you not provide, if you not provide aseptic condition, what will happen? If you not provide an aseptic condition, entire culture media contaminated, entire culture media contaminated. That's why the tissue culture does not grow. That's why we need to provide aseptic condition. In aseptic condition mean the conditions free from the any type of microorganism, any type of pathogen, so as to prevent the loss experiment by the contamination. We need to provide this so that we save the experiment from the contamination. For the sterilization, that means the complete removal of killing microbes. The most common contaminants in cultures are fungi and bacteria. The mostly the culturing, the culture media infected with the bacteria and fungi, especially in fungi, rhizopas, mucor. So that's why the rhizopas called as a laboratory weed. So like that other bacteria also, most of the bacteria also contaminate the culturing process. Okay, right. Measures to be taken maintaining aseptic during the aseptic condition during the tissue culture area. First of all, the sterilization of the culture vessels using the detergent and autoclave. So culture vessels in sterilization process, let me draw here. So what are the requirement in sterilization, tissue culture, vessels, mainly vessels, tissue culture tube or tissue culture tube vessel, that means tissue culture tube, TTP, tissue culture tube, their requirement. The first of all, our tissue culture tubes need to be using detergents, autoclave. First of all, wash with detergents. Even even after washing the detergents, you should keep on autoclave. Autoclave look like a pressure cooker. After 15 minutes, if any microbes present, they are eradicated. And apart from the test tubes, and we frequently used in tissue culture process, some of the instrument like forceps we use for uh, holding the explant. Needle also we need. So this two forceps and needle, as it is a minute structure, the flame it. The both are flamed with help of heat. If any, if any microbes present on surface of the forceps and needle, they should be killed. Next, sterilization of culture media using Fetal sterilization autoclaving method. That is the best method. If finally we keep in autoclave for 15 minutes. Okay. Now these are all for the instrument preparing for culturing. So vessels, test tube, tissue culture tubes, forceps, needles, and also you should wash your hands properly. So they are the main. So what are the other requirement? Explant also need. Explant also we should sterilize. The next one, explant sterilize. Because already told you the explant collected from the outside the plant. Outside the plant. This is the plant. From the plant outside, explant, we separated the explant. So where is the plant? Plant is the outside, not lab. The plant is outside. So as it outside, it is mainly infected with most of the microbes. That's why we need to sterilize the explant what you separated from this plant. For explant sterilization, uh, we utilize the some surface sterilization explant using surface disinfectant like silver nitrate, silver nitrate 1%, silver nitrate 1%, and H2O2 10 to 12 percentage, and to H2O2, bromine water 1 to 2%, sodium hypochlorite, sodium hypochlorite. These are the major chemicals, sodium hypochlorite solution. So we should sterilize the explant AgNO3, H2O2, or sodium hypochlorite, silver nitrate, hydrogen peroxide, sodium hypochlorite. With help of this explant also sterilize. So now we sterilize the explant for growth of the plant, culturing for the culturing substrate, and this culturing explant we keep inside the vessel, tissue culture vessels, with help of the forceps and needle. That's why here the forceps and needle, these are all need to be sterilized, right? So anyway, here the all the instrument to utilizing tissue culture and explant, they are all sterilized. Now sterilization process also completed. Sterilization process also completed. Now clear. So now in this first of all, in first step we prepared the lab. We opted the proper optimum lab. After that we prepare second this one. So this is second. We prepare the culture media with help of the micro and macronutrient and sucrose and vitamins and amino acids. Next, we create a aseptic condition for the instrument and aseptic condition for the explant. This is old concept about the plant tissue culture. Now let's take the steps in plant tissue culture. In examination point of view, if they ask about write the plant tissue culture technique, you should write all these characters. And also now I will going to talk about the steps. Steps also you will have to write. And in examination point of view, if they ask about totipotency, write the totipotency characters. If they ask like that, you write the same answer. What is totipotency? What is totipotency of actually why the plant cell have the capability of regeneration? Because of totipotency property. The totipotency property means the plant cell can able to have the divide, the regeneration capacity. That means the vegetative cell or any cell, any plant cell have the capable of division. That capability called as totipotency. So in examination point of view, if they ask about write about totipotency or tissue culture technique, the same, right? Now, let's see the steps. The basic steps for in vitro culturing plants are, is it clear first? Next. 1 minute. Just a minute. Now let's see the steps in general techniques in plant tissue culture. Uh, Actually, in first step, selection and sterilization of explant. In first step, first selection and sterilization of explant. That is the first step. First, we have to select explant. Where is from explant? Explant from the outside the plant. So now the intact the explant, this excision of explant and proper trimming of explant. After that, we need to surface sterilization. Already you know how the surface sterilizing. How the surface sterilization? The surface sterilization with help of the silver nitrate, already told you, with help of H2O2, hydrogen peroxide, sodium hypochlorite. These are the chemical using for the sterilization, surface sterilization of explant. After that, the sterilization of glassware. So in this process, sterilization of glassware also we need. So the sterilization glassware with help of the autoclave process. and explain washed and sterilized distilled water. Right? So one is completed. Selection, sterilization explained now. The second step is preparation and same sterilization, preparation, sterilization of culture medium, culture medium. That is the second one. So second culture media preparation also just now we completed. Just again one sec. Uh, Culture media. First of all, the culture media preparation. How the culture media preparation? Already told you for preparation culture media, they need agar preparation culture media. Let's take preparation culture media, agar, micro, macronutrients, vitamins, hormones, hormones, sucrose solution. They are required for the sorry for the preparation. Now the sterilization process. In sterilization, culture media after prepared culture media, the entire culture media keep inside the autoclave. Now what happened? What are the requirement glassware instruments sterilized? Here the explant sterilized. Now third step is so first step completed and second step completed. Clear? So in first step, what happened? Selection of explant and sterilization. One. Second, preparation and sterilization of culture media. Right? So in first step, what we selected, a suitable explant is selected and is then excised from the donor plant, and explant is then sterilized using the disinfectants. And next one, prepared the preparation and sterilization of culture media. A suitable culture media is prepared with special attention towards the objectives of culture and types of explant to be cultured. Prepared culture, prepared culture medium is transferred into sterilization, sterilized vessels. Already vessels also sterilized here, then sterilized in the autoclave. Now the inoculation. Third step is inoculation. Third step, let's change color. Third one, inoculation. Sorry. Third one, inoculation. inoculation of explant in the culture media under aseptic condition. This is also under aseptic condition. This is very, very important. That means, what is inoculation? Inoculation means entry of explant into the medium, into the medium. That is called inoculation. So what happened in inoculation process also? Already in first step, you sterilized explant and sterilized already medium. But even though the both are sterilized well, in during inoculation process, that means in when you uh, inoculated inside the media, in that time also there is a chance to infect with microbes when this process you conducted in outside, not in aseptic condition. If you perform in a non-aseptic condition, there is a chance to attack the microbes. That's why they need to the carried out in the specialized chamber. The chamber called as laminar airflow. That is called as laminar airflow. The chamber called as laminar airflow. I will write. The chamber called as is carried out inside the laminar airflow. This laminar airflow create a aseptic condition. It produces the UV light. So due to this UV light, if any microbes present, the microbes would be killed. Right? Now, so culture are then incubated. So once again recall the sterilization explant is inoculated. The inoculated nothing but the simple transferred on the culture media under the aseptic condition. This aseptic condition created inside the laminar airflow. That's why this is performed in laminar airflow only. Now the after thought, the fourth step is incubation. Now this one. Sorry. Incubation under the suitable condition for proper culture. So cultures are then included in the culture room where appropriate condition of light, temperature, and humidity are provided for successful culturing. So that is the importance of incubation. It is incubated in laminar airflow 5 to 6 weeks. What happened during this 5 to 6 weeks? During 5 to 6 weeks, explant can absorb the minerals, vitamins, sucrose, utilize the sucrose also. Now it is trying to develop it into a callus. That means trying to develop it as a growth callus. So these are all happening in during the incubation period. After that, we subculture. Subculture means the cultured cells already due to this waiting period, incubated period, the callus is formed. The callus means undifferentiated part. The mass of group of cell formed. Now we have taken the cultured cells, now transferred into fresh nutrient medium to obtain the plantlets. Now after that, so after that what happened? In a fifth one, subculturing. Subculturing. Desired characters. So after getting callus, now this callus subculture. After subcultured, now first, last one, planted transfer. After the herding process, that is acclimatization of plantlets to the environment, the plantlets are transferred to the greenhouse in a pots. So that is so anyway during subculturing the small plantlets are get these are very sensitive. This tends to the transfer into polythene bag and finally the transfer into the greenhouses. Greenhouses, right? So these are the techniques. These are the plant techniques. So after that, after greenhouse, the transfer into the outfield. Okay, transfer it planted in outfield. That's it. So this is the main steps in plant tissue culture, right? Now, so clearly once again, in steps, first of all, this total, this one first step from the that means selection of explant. Selection of explant. In first step, what happened? The selection of explant, which means the explant, where is explant already the plants grow outside. So we have to select the explant from the outside of the plant. Uh, so the suitable explant is selected and is then exercised for the donor plant, is properly trimmed with the explant. Next sterilization. Next sterilization of the explant. Already told you the explant sterilization, silver nitrate, H2O to NOCl. So that is the first one. The second step, preparation and sterilization of culture media. Preparation of culture media, hormones required, agar-agar required, some of the antibiotic required, micro, macro, vitamins, they are all required for the preparation media. After both sterilized and prepared, now this both inoculated. This the third one, inoculation. Inoculation means transfer of explant into the culture media. That is inoculation, which is done in laminar airflow, which creates a aseptic condition to inoculation process. Now incubation period. Period means growing. That means under the suitable condition, what happen? All the macro and micronucleus taken by explant and try to grow as a callus parts. After grow callus parts, we separate the callus and subculture again. During the subculturing, small plantlets are formed. Now the plantlets, the planted in polythene bag and keep under the greenhouses. After greenhouses, after weeks, now transfer into the outfield. Outfield. So these are the basic steps in uh, tissue culture, right? So this is the most important for essay question. And not only for essay and remaining all culturing process. All culturing may be the organ culture, protoplast culture, any other type of culture based on this technique, based on the same steps. That's why this is very, very important. If you not get understand about this, you can't get the further classes. You can't understand further classes also. Right. Now let's see the applications of plant tissue culture. This is also another important application of plant tissue culture. Also important here. Right. So first application is micropropagation. What is micropropagation? Micropropagation means micro means small. Propagation means number of plants formed. That means one plant to another plant, another plant to another like this. More than uh, one plants formed within a short period, that is called micropropagation. Just a bit. Right. So in micropropagation process, tissue culture helps in this rapid propagation of plants. That means what happened here? The plant population derived from the single donor plant is called clone, and multiplication genetically, these genetically identical copies of that cultivator is called clonal propagation, which may be useful tool to get large population of plant species having a desired traits, desired character. In tissue culture, from callus mass, a large number of shoot meristems can be regenerated within very short time, already told, within very short time and space. As a result, large number of plantlet can be produced from the such callus tissue. The most obvious advantage of the technique is the large scale production of plant same genetic stock. That means, for example, here the only one plant there. From the one plant, you selected the different type of explants, explant, explant. After sterilizing, these are all incubated in this. That mean transfer into the medium, transfer into the medium. Medium first callus formed. No, you subculture these callus variously. This is the callus. You separated some portion, some portion. Now this group of cells you have taken the 10 subculture. This 10 subculture gives the 10 plants. This is for the basic understanding. So what happened? What happened here? This donor plant. How many donor plants here? Hello? Donor plants only one donor plant. This one donor plants produced number of group of plant population. This is called micro propagation. This is called micro propagation. Right? Next one. Two somoclonal variations. Soma, right. That's somoclonal variation. So the variations generated by use of tissue culture cycle, that is called as somoclonal variations. The scientist name Larkin. That means what happened during the micropropagation? In micropropagation, one plant to get number of plant species, number of group of plant species generations. But these plants, the same genetic material copy paste. What the mother plant have the genetic material, the same genetic material present in this population also. But sometimes what happen, whatever it may be the reason, due to some reasons, the variations occurs. Some variations. That means there's some gene structure modified during the culturing process. So these variations occur during tissue culture. That variation called soma clonal variation. Why it is called soma clonal? Actually, the variations that means the gene changes occurs in reproductive cell, but not vegetative in general. In reproductive cell, in the form of crossing over, they can exchange the genes one to another. But in tissue culture, in vegetative cell, the variations occur. That's why it is soma clonal variation. So this genetic variability is due to the cells of various ploidy level, maybe ploidy level increase or decrease, and genetic constitutions of the initial explant are also maybe developed due to the different cultural conditions. This kind of variation may show some useful character, may show but not always. They may show the useful character. These useful character not present in mother cell, may not be present in mother cell, but that such condition obtained in the coming offspring. That coming offspring means culturing plant. The mainly the beneficial characters are resistance power to a particular disease, herbicide resistance, stress tolerance. They are all they are all the get beneficial. So this is called somaclonal variation. But remember, the somaclonal variation may be defined as those variation which occur in the cultured cells, all that only cultured cells or tissue. The plant regenerated from the such cells in vitro. The somaclonal variations are usually heritable for qualitative as well as quantitative characters of plant. That means this is the culture plant. From this explant, you selected number of plant. But while the culturing, there is a changes are there. But these changes, when you fertilize with these two plants, the changes also they carry forward to next offspring also. That is important thing. And some of those crop species such as potato, tomato, oats, wheat, rice, maize, datura, soybean, we have found that somaclonal variation. In simple word, the variability which is generated by the use of tissue culture cycle is termed as somaclonal variation. Soma clones is general term which use desirable those plants which have been derived from the type of somatic cell culture. So I already told you this may be useful to the plant, the useful nature, the resistance, particular disease or herbicide, instead, whatever it may be, the stress tolerance also. They are the benefits, right? Next one, another application is somatic embryogenesis. How the embryo form in general, somatic embryogenesis. Somatic embryogenesis, before that, you should know embryo formation. Already, just now I told you the embryo formation by zygote, the zygote fusion of male and female gamete, both fused to formation zygote. This generally formed occur. The somatic embryogenesis may be achieved from the embryonic cells of the direct explant or the embryoids developed within the callus. tissue culture from induced embryonic cells. So the somatic embryos achieved from the embryonic cells from the direct explant, from the direct plant. That is one method. But is another one is after what happened during the culturing process, the culture formed. This is the culture callus. For example, this is the callus. From this callus, when you induced as an embryo from this callus, from this group of cells, callus, embryo, let's change color. Embryo, this group of embryo also develop. Actually, this embryo developed from the callus, from the vegetative cell, but not reproduction. So this is called, this embryonic cell called as a somatic embryogenesis. This technique, this application of this technique is the mass production of adventitious embryos. Not only one embryo, number of embryo formed, which ultimately develop into complete plant. That means actually one embryo develop one plant. If multiple embryo form, it produces the multiple plantlets. These somatic embryos can be encapsulated with suitable nutrient containing this is encapsulated this somatic embryo for future use. We can encapsulate that means coating, okay, coating with help of some of the chemicals, sodium alginate, potassium alginate, right? For the protection, we can encapsulate it with these chemicals. The somatic embryo, this is called somatic embryo process. And somatic hybridization, sorry, somatic embryo, where it is, just wait. Uh, and the somatic embryos also called as a synthetic seeds. Synthetic seeds, there is also called synthetic seeds. There are living seed like a structure which are capable of giving rise to plant when shown in the field. An artificial seed made up of somatic embryo, that means encapsulated with protective layer. Already told you the gel, sodium alginate, potassium alginate gel. So this is about somatic embryogenesis. And another important one is another application, secondary metabolites. How the secondary metabolites, how the primary products formed. What are the primary products? The primary products, carbohydrates, protein, the carbohydrates, proteins which are formed by photosynthesis. By photosynthesis, during photosynthesis, what happened? Primarily food material formed. That is called primary metabolites. How the secondary metabolites formed? The secondary metabolites formed after utilization of the primary. After you utilize the primary one, now it is develops as a secondary metabolites. The remaining products convert into secondary metabolites. What happened if you just compare with animals? In animal, first we have taken the actually we can't prepare the food material. We take the food material from the outside. So after the utilization, the carbohydrates, these carbohydrates utilized for the growing for energy production. What happened? Excess material. This excess material convert into uh, secondary metabolites, amino acids, urea, like that. Now they can excrete it out in the form of urine or desiccation through the excretory system. But what happened in the plant? In plant, there is no excretory system. So that's why the secondary metabolites, whatever secondary metabolites may be the alkaloids, flavonoids, whatever it may be, they are all stored inside the plant. stored inside the plants. Just right. During this, I think clear primary, secondary by photosynthesis. Primary form after utilization the plant body, the primary converted into secondary and stored inside the plant body. They may be alkaloids, flavonoids, alkaloids, so on. But these form of waste material excreted in animals, but not plants. Plant storage. So these are the beneficial to the plant body. So the production of may useful compounds like alkaloids utilized for the plant body, vincristine, quinine, steroids, glycosidic compounds, essential oils also, coloring agents. They are all secondary metabolites, and they can be done by plant tissue culture. The aim can be achieved by the selection of the specific cell producing the high amount of desired compound and develop of a suitable medium. For example, you want to get the more alkaloid vincristine, quinine, vincristine. It is the cancer, prevent the cancer. To get this vincristine, you select Vinca rosea plant because the vincristine present inside the Vinca rosea. You selected the Vinca rosea plant for the explant. After you selected the explant, these plants can get in mass production. That means you can get the number of secondary metabolites, number of vincristines. In general, secondary metabolites produced in plants cell culture are rather the small in amount. Of course, in culturing also produced, but in small amount. But by clonal selection, particular high-yielding clones of cells can be isolated. Sometimes the plant's culture may provide the helpful way for production of secondary metabolites by feeding the culture with expensive product precursors or by manipulating their biosynthetic control mechanism. So this is about secondary metabolites production. Next one, in vitro morphogenesis. Actually, it is not part of our syllabus. Types of culture. I will explain one by one later. First, the types of culture. Let's see here. Process same. Culturing technique same. Cell culture, organ culture, callus culture, protoplast culture. These are all. So in this, almost all same techniques, but cell culture is separated cell. Organ culture, we after the organ. Callus culture, after get callus, you subculture. Protoplast culture, I will explain the one by one. Yeah, among this, the most important one is protoplast culture. This is very, very important. Protoplast culture, very, very important. First, the cell culture. First, cell culture is the culture of isolated individual cells obtained from the explant tissue or callus, from separated from the explant tissue or callus. Explant tissue made up of cells. That means you directly selected from the cell of this tissue. or after get the callus during the tissue culture process. After get callus, from the callus also you can select the cell. These cultures are carried out dispensation medium but not solid medium. This is separated medium, dispensation medium, and referred to as suspension culture. This is called suspension culture. The organ culture is a culture isolated plants organs like embryo, seed, root, endosperm directly, anther, ovary, ovule, meristem, nucellus, callus. Not this one. And callus culture involves the cultural differentiated tissue from the explant, which differentiated into in vitro form the callus. Now the protoplast culture is that culture protoplant protoplast then laboratory. I will explain clearly later. This is just brief introduction about this. Right. In detail, let's see the cell culture process, type of cell suspension culture, broth culture, continuous culture, synchronization suspension culture, chemical starvation methods. Methods assess the viability cells in suspension culture like that. Just wait. Right. Cell culture. So already told you for the culturing of the cell, we don't use MS media, solid media. We use only suspension culture. That is cell suspension culture. Let's take pen. Cell suspension culture. Simply multiplying the single cell at higher rate in liquid medium. The medium is liquid. Already told you the culturing cell culture, we utilize the liquid medium that is called suspense culture. The liquid medium continuously agitated, continuously disturbed, shaked, and orbital shakers. It is also popular tissue culture method for many industries to extract certain components of the plant cell. What happened? For example, these are the shakers. On the shakers, you take the plant cell in liquid medium. Liquid. What happened due to the shaking? Due to the backward, forward, and lateral movement of shaking. Due to shaking, the liquids and cells all together mixed, right? So this agitation, the medium also exerts the mild pressure due to the shaking. The pressure create exert pressure create on tissue, breaks it into smaller species and single cell. That is our first concept. We have to separate the cell. How the cells are separated when the tissue or collars are placed in the shakers. Due to the shake, the cells, due to the pressure, the cells are separated. This agitation helps in maintain the uniform distribution movement of the cell in the medium. That it is an important procedure for cell suspension culture. Actually, why do you need to provide constant agitation to medium? How do we get the single cell callus? The callus is an irregular mass, right? Of cell plant tissue, do not have any structure, but possess high ability to differentiation into the different plant organs. We can obtain different calli from single explant. However, this calli from the same explant might have show the variation in color, texture. These calli may be compact, free bole, and texture, and it is in dark and color. You need well-established friable callus culture for your cell suspension method. One of the interesting facts about the callus culture is that you can multiply them for unlimited period, unlimited period of time without any specific differentiation. You can do this by periodically subculturing on fresh medium containing phytohormones such as oxygen cytokinin for cell division. Now, Those, let's see the types of cell suspension culture, broth culture, and continuation culture. Batch culture. Batch culture. First of all, batch culture. The batch culture is grown. Batch culture is grown in a fixed volume of nutrient culture medium. In this culture, cell divisions and cell growth coupled with increase in biomass occur until one of the factor culture environment become limiting. Right. For example, already we added the some of the nutrient. So of course we use the micro, macro, vitamins, hormones. They are all. But while the culturing process, any one factor act as a limiting in this batch culture. This cell exhibits the following five phases growth when the cell number suspension culture is plotted against y-axis and x-axis. So if we take the graph, log phase, linear phase, declaration phase, stationary phase. The first phase, cell division, cell manipulation highest. For example, if you take like this, slow cell division, highest number increasing, linear slow, decrease, stable, stable. Log phase, just cell division start. LOG log, this is also log phase, where the rate of cell multiplication is highest. That's why the growth highest rate. It is press slowness again after growth, again slowness in cell division, increase in cell size. Stationary phase means equivalent is constant. Declaration means decrease in cell division and cell expansion. Now constant, later decrease, gradually decrease. Stationary phase represent the constant number of the cells, their size. The batch culture can be maintained by continuous transfer of the small amount of the suspension medium, continuously transferred. Medium containing inoculum, the fresh medium. Regular intervals, approximately 2 to 3 days. That means already you added the culture medium. After that, you utilized all nutrients. One of the nutrients act as a limiting factor. Then the growth retarded. Then again, when growth stops, when again you have to transfer the another medium. In regular interval, you will have to added, re-added in regular interval. Then only the plant grow, the tissue culture grow. And next one, so the batch culture, it is a closed system culture. We can grow the cells in fixed amount of culture medium under the appropriate condition. This kind of cell suspension is performed using flask volume of 200 like this. Conical flask, conical flask in this taken liquid medium, colors, cell separated while in orbital shakers, used in shakers. You can treat the first flask, the cells inoculation, further flask suspension. That means already you added in this nutrient, already all nutrients are there, but the growth occur, but stop the growth. When the growth stop, and again transfer it to the another flask, another culturing media. And again stop, after some time again stop growth, again transfer into another media. So this is closed. This is called takes place in closed circulation. The one major, let's see the some of the demerits. One major drawback of the batch culture is that cell grow Up to a certain point, already told you, the certain limiting factors. Then the cell growth becomes stationary. In the stationary phase, the number of cells and size cells remain constant. The cell reaches stationary phase either because of exhaustion of some growth factors and accumulation toxic metabolites. Any other act as a limiting factors. However, you can avoid this phase by providing fresh medium. That means, of course, already told you, when it is retarded, again the fresh medium, again added fresh medium. It is doing continuously in regular intervals. So this is about batch culture. The next one, continuation culture. Let's see the continuation culture. Continuation in this type of cell suspension culture, you can maintain the constant phase of cell growth. No decrease, increase, constantly maintain. If the fresh medium is continuously added and the leftover nutrient. First medium, what happened here? In first one, it continuously added. But here, continuously added and first one removed. In this constant phase cell growth, here the fresh medium is continuously added and leftover the nutrient and metabolic produce are constantly removed from the medium. First of all, this is first medium utilized leftover and add new medium and again leftover and again new media and again leftover and new media. This is happened. Thus, by using this method, you can avoid detoxification to the media. Hence, the overcome drawback the batch culture. What about batch culture? Batch culture already nutrient media added, stop growth, and again nutrient media added, sometimes grow and again stop like this. But in this type, what happened? There is the toxins are formed. There is chance to formation toxins. So in this continuation, two types continuation culture, chemo starts and turbo starts. In chemostat, in this method, you can add the fresh medium while harvesting the same amount of the culture. This maintains steady cell growth. However, you need to adjust the concentration one of the nutrient nitrogens. In this turbinates, in this method, biomass of cell continuously maintained at the level of below the maximum heat. You can provide the cell nutrients excess, unlike the one limiting factor in chemostat culture. You can control the additional of the fresh media by observing the turgidity of the culture. So this is about the continuation culture. Next one, synchronization suspension culture and chemical methods, starvation methods, and callus culture. I will complete tomorrow. There are callus culture and factors of callus culture. I will complete tomorrow.